Now, the topic of my talk is on the role of subclinical infection in disc degenerative disease. Now, we all know that disc degeneration is inevitable, but the etiopathogenesis is still a controversy. And the big question mark is on the role of subclinical infection. Now, it all started with this paper in 2001, where Alistair actually demonstrated the presence of bacteria in disc samples from herniation. And this factor got augmented by the paper from Albert, where he showed good results following antibiotic therapy in a certain group of low back pain patients. But this idea is still a raging controversy. And most of the non believers actually strongly believe that the bacteria that is demonstrated in these studies is actually a contamination. But however, there is a strong proof that bacteria can be an initiator and propagator of many diseases. Like, for example, we know H. pylori is the cause for peptic disease. We know that there is a strong association between P. acnes and adhesive capsulitis. And in rheumatoid arthritis, there is a whole amount of organisms which have been proven to initiate and propagate the inflammation. And in many cases of aseptic loosening, which were previously thought to be cement related, are now known to be infection. In 2017, we also presented this paper on the Easels Prize winning paper on the role of possibility of subclinical infection being a cause for disc disease. We not only demonstrated bacterial proteins in normal discs, but to a high level in discs with disc herniations and disc degeneration. But there is still a lot of controversy, and this controversy is between infection and contamination. Now, if we have to settle this, there are many missing links that have to be addressed. And I would like to present our work on this subject. First of all, we need to document the presence of bacteria in the discs. We need to decide whether it is contamination or actually infection by showing viability of these bacteria and that there is an infective process going on, and this is the cause for disc disease. So it all starts with the primary question: does normal disc have bacteria? Now, most of us strongly believe that the discs are highly sterile in health because they are the largest avascular structures in our body. But our understanding of infection etiology has always depended on our ability to identify organisms. So far as we restrict to culture-sensitive methodology, we will miss identifying a whole lot of microorganisms which cannot be cultured easily. And so metagenomics and next genome sequencing is the future. And this allows us to identify all the bacteria by DNA sequencing. And this is what we employed. And we looked at 8 MRI normal control discs, 8 degenerated discs, and 8 herniated discs, which were all harvested under sterile precautions and immediately snap frozen. What we found was that there was a rich bacterial presence in all 3 groups, and even in the normal discs, which showed that even in the health, normal lumbar disc has a good presence of bacteria. But the microbiome of normal discs differed from that of the degenerated and herniated discs, both in biodiversity and abundance. While normal discs had a wide spectrum of bacteria and relatively very low abundance of established pathogens, degenerate discs had a reduction in the spectrum, but they had a relative increase in the number of pathogens. Beneficial bacteria like Bacillus coagulans and Bacillus clausii, they were all present only in organ donor discs. And Lactobacillus mucosae and Sphingomonas were present in abundance only in the organ donor discs. Whereas bacteria with pathogenic potential like Pseudomonas fragile, Privatola, and Streptococcus and Privatola pallens were all present only in the degenerated and herniated discs. And this is what we reported last year in the European Spine Journal, that human intervertebral discs harbor a unique microbiome, and dysbiosis determines health and disease. Now, what is meant by dysbiosis? Dysbiosis is the balance in the normal between the type and quantity of bacteria which is present in a tissue, and an alteration of the normal ratio can actually determine and cause a disease. So we have symbiotic bacteria, commensals, and pathogens. And when they are in balance, it is the state of health. And an imbalance between the type of organism present can actually cause a dysbiosis, which can result in a problem. Now, in the last 2019 NAS Outstanding Paper Award, we established that there is an inflammaging, a constant low level of inflammation occurring in diseased discs. And we also showed that this could be because of a bacterial induced inflammation. Now, if you looked at normal discs in healthy young discs, most of the proteins that were present were structural proteins and metabolic proteins, and there was no complement cascade proteins or degradative proteins. Absence of inflammatory markers and degeneration was in the young and normal discs. Contrast to this, in degenerated discs, we found an upregulation of complements and degradation proteins and inadequate complement inhibitors, which actually showed that this inflammation which is present in the degeneration and herniation could be as a result of bacteria. In the third part of our study, there was further proof because we established the presence of host defense response proteins. Now we know that whenever there is a pathogen exposed to the tissue, the human body mounts up a defense. And the proof of this can be found from the presence of human defense response proteins. Now, you could see here, we were able to establish a whole number of host defense response proteins. For example, we found that prolactin-induced protein, it was 9 times higher than what was present in the normal disc. And we also know that these act as the first line of defense against invading pathogens. Similarly, complement component and heat tolerant. There was a 3.6-fold variation. And we know that these are all a part of membrane attack complex on bacterial cell membranes, in which beta subunit helps in complement-mediated bacterial clean. Proof of all presence of these bacteria actually stand as a proof for infective pathway and activity going on. For example, defensin was also uniquely present only in disc degeneration and in herniation. And defensin is a family of antimicrobial and cytotoxic peptides found in the microbicidal granules of neutrophils that helps in phagocyte-mediated host defense. And the presence of this and another 42 host defense response proteins in diseased discs actually showed proof of inflammation, and an inflammation that is initiated and progressive because probably of bacteria and infection. So we came up with a unified pathway. Most of us used to think that the traditional hypothesis that degeneration is mechanically induced, but we do think that this is an important factor, and this can actually be the initiator of a cascade of events. Mechanical disruption can either lead to end plate breaks or annular tears, both of which lead to neovascularization of the disc. There is ample proof for this in literature. Neovascularization then carries the bacterial, and this can lead to bacterial inoculation, and that can lead to a low-grade infection. And this low-grade infection can lead to a constant source of inflammation, and this inflammatory cascade and perpetuation of inflammation can lead to disc degeneration and herniation. Now, there can be a genetic susceptibility for this, and that is the way that genetics also will play an important part in disc degeneration. So our study over a period of time has shown proof that bacteria is present in even health, and they are present in a difference of ratio with dysbiosis in the degenerated disc. It is not a contamination because we have proved that the virulence, that they are multiplying, they are viable, and actually they are a cause for infection because we have also proved that there is dysbiosis and there is inflammation and the presence of a host defense response proteins, which actually shows an infective process. We know that human cells versus bacteria in the body, we are outnumbered by the bacteria by a ratio of 10 is to 1. Many of the bacteria which we thought were commensals, now we know that they can be a source of many kinds of disease. Now, it was Chanakya who actually said to underestimate the enemy and to mistake an enemy as a friend are the two mistakes that any king must avoid. So I think in the recent few years, we have now come to the state, mainly also because of the fact and the ability of us to identify all the bacteria that are present in the tissues by next genome sequencing, that many of these bacteria are the source of a constant inflammation in the tissues, and this is also probably the cause or at least one of the causes for disc degeneration. Now this is will be very important because of the role of bacteria as it is being established in health and disease, and if we can prove that there is a role for infection in disc degeneration, it will dramatically change the way we look at this pathology, and it will also offer and open up new possibilities for treatment of these. As Louis Pasteur said, we are all on the verge of mysteries, and the veil is getting thinner and thinner, and hope that the truth of what is the real pathology behind disc degeneration will be in our hand very soon. So thank you very much again for this opportunity, and my greetings to all of you.
Actually, this was a very, very interesting lecture. And the next lecture is even more interesting. It's on Modic changes, what they mean for the surgeon and the patient.
Thank you very much for the opportunity to discuss our work on whether Modic changes are a predisposing factor for surgical site infection. We have no conflicts of interest. Now we know that surgical site infection, especially in the spine, has great socioeconomic morbidity and mortality. There are very strong evidences for many of the predisposing factors, and many of the factors have a moderate association. These are very well known and are common to all surgical operations. However, now there is a growing interest on whether the end plate changes and erosions, which were first described by Modic in 1988, is a predisposing factor for infection. We also know that the end plate erosions can be accurately scored and measured by the total end plate score, which was described by us in 2008. So our aims and obstruction and a high TE plus scores has independent risk factor towards postoperative infection. The study design is a retrospective case control analysis with strict inclusion and exclusion criteria. And it was done in 2016, 17, and 18. Of the 8,253 surgical procedures at that time, after inclusion and exclusion criteria, a total of 1,124 cases were compared. In all these cases, the type of Modic change, whether it's type 1, 2, or 3, or a mixed Modic change, and also the location and extent of the Modic change were documented. We also scored the extent of involvement of the end plate by the total end plate score, which gave a score from 1 to 6 for each of the end plates on either side of the disc, with a total possible score of 12 for each disc. Now the data collected was the type of surgical infection, whether there was a tissue positivity, and also looked at all the comorbidities. And appropriate statistical methods were utilized. Now, the incidence and surgical site type of surgical site infection was correlated to all the other factors like age and sex, but also the Modic change and total end plate score, and to the presence of comorbidities and what was the culture yield possible. Now, it is seen here that age and sex did not have any significant difference between those who had infection and who did not have infection. Now, 59.6% of patients, 670 of the 1,124 had Modic positive, and a majority of them, up to 82%, was type 2. 44 patients, 4% of these patients had infection, and they were cases, and they were compared with the 96% who were controls who did not have an infection. Now, it is very clearly seen from here that Modic changes had a high association with a surgical site infection, highly significant. And there was no correlation between the distribution of the Modic change or the location of the Modic change or the extent of Modic change. So this was very important. Just the presence of a Modic change, irrespective of the type, and irrespective of the location or the extent, seemed to be a risk factor for surgical site infection. There was also no difference between superficial and deep infection. And we also looked at the second important score of the total end plate score. And here too, we found that whenever there was an erosion of the total end plate score, there was a highly significant correlation with the infection. Now, whether it was for a single level surgery or whether it was associated with Modic or not associated with Modic, whenever a total end plate score increased more than 6, it was found to have a high incidence of infection. The maximum correlation was for discectomy and not for fusion, decompression, or decompression and discectomy. And this we have also found in our practice that whenever you do a discectomy in Modic change patients, there is a poor outcome and also a higher state of surgical site infection. Multivariate logistic regression analysis also showed that the odds ratio for an infection was highest for a total end plate score of greater than 6, with the next highest score for Modic changes at 2.7. And the presence of other comorbidities like diabetes, hypothyroidism, and a high BMI, they all scored much lesser than these two entities. It is also very important to note that only 25% of them actually grew a culture positivity, whereas 44% of them were culture negative, and hence they had to have an NGS to have a proof. So Modic change, that it is associated with the low-grade subclinical infection, has been discussed in many different papers so far, but most of them were basic science and systematic reviews, but there has been no major studies on whether there is a direct correlation with postoperative patients. Our recent papers on this to prove that there is an inflammaging whenever there is a Modic change, and also when our insight and full analysis on his infection, the possible initiator of disc disease, they showed that there may be a possible pre-existent infection whenever there is Modic change present. And this study actually proves that the presence of Modic change, irrespective of the type, or a higher total end plate damage score of greater than 6 are independent risk factors for infection. And when you operate on such patients, it's important to have a good preoperative counseling of these patients, careful postoperative observation, and we need further studies to know whether we should do an extended antibiotic regime in these patients. Thank you very much for this opportunity, and we would actually thank you very much.